Hi biologists. Today we're going to be looking at transcription, which is one of the processes in protein synthesis, also known as gene expression. In front of you are your learning intentions and success criteria. So make sure you have a look at that so you understand what you need to learn and how you need to demonstrate it. So firstly, I mentioned that word gene expression. Anytime you hear the word gene expression, it just means that we're looking at the genetic code, copying down a specific part of it, a gene, and then we're going to transform that gene into a functional protein. So we're expressing the gene. It's the same as protein synthesis, made up of two processes, transcription and translation. Today we're going to look at transcription. Rightio, this is transcription in a nutshell. So let's have a look at what we can see. Firstly, our familiar DNA strand here, still coiled up and connected by those complementary base pairing weak hydrogen bonds here. We've got a section of our DNA molecule. This is the gene. This is the bit that's going to be copied or transcribed. Now you got a bit of a funny looking blob of a few things here. This is called a transcription factor, and I'll discuss that in a little bit. But essentially this tells you where the RNA polymerase is going to attach to the start of the gene, tells the enzyme where to start copying, basically. And that's what happens. So once RNA polymerase attaches, then we get some unzipping and unwinding of the DNA molecule, and RNA polymerase works its way along the template strand of the molecule and adds bases, RNA bases this time, complementarily to that template strand, so we get elongation in this direction here. And then as that RNA strand elongates, the back of it kind of pops off and the DNA reconnects and winds back up, and this happens all along the template strand like that until the whole gene's been copied and we end up with our RNA single strand over here ready to go and get processed. A couple of things to note here. We've got two strands called a coding strand. So this coding strand will have the same bases as the RNA strand, except for, obviously, RNA will have a U instead of a T. That's uracil instead of thymine. And the template strand is going to be the complementary strand to the mRNA. And that's important for a little bit later down the track, and I'll mention that when the time comes. Okay, so I mentioned transcription factors. This is an illustration of what that looks like. There's more detail here than what you really need, but the main point here is that we have our DNA strand and we've got lots of proteins involved, all right, lots of chemicals involved, and what that does is it tells the RNA polymerase here where to bind and then start production of that mRNA strand. So this idea that substances can control when genes are being expressed is really important, because cells can't express all of the genes all of the time. If they did that, then they wouldn't be different from one another. So some cells in our body will express different genes to others. In fact, they will all express different genes to each other. And there are chemical messengers that tell the cell when that happens, and those chemical messengers are these transcription factors. So these are important, and they'll come back into play a little bit further on in the course. Right, so in a nutshell, here it is. We're producing messenger RNA in transcription. The RNA gets processed after it's been transcribed. So this word here, transcription, actually means to be copied down. So we're copying down the code here, and you can think of RNA polymerase if the mRNA strand is the copy of the code, then the RNA polymerase is the scribe that does the copying, right? So we end up with this copy of the code that then needs to be processed so we can make full sense of it. And what that means really is that introns are removed and the exons are remaining. They're the ones that are expressed. And then that leads to the mRNA being ready to go to make polypeptides, so it will then move out of the cell ready for translation. If you need a bit more detail on that RNA splicing, go back and have a look at these student understandings here, D7.1 and 7.2. And finally, here are some summary notes for you to take down into your book. So now that you've had a look at that, let's go through a past exam question. So the question is, describe the process that occurs in the nucleus of a human cell that enables the protein telomerase enzyme to be produced. So there's a couple of things to note here. We're given a name here of an enzyme or a protein, telomerase enzyme. Now, you probably don't know what that is, and sometimes you're going to get questions with words and names in it like that you don't know what it is. What you might note is this suffix here, the A's, indicates that it's an enzyme, and here we've got an enzyme here. It actually says it's an enzyme, and we're going to learn about them next. So there's a bit of information that you need to actually extract here, and we've put it in bold for you here, but that won't happen in an exam. So the process that occurs in the nucleus of a human cell, well, that's the giveaway, and the process is transcription. That's the only process within the nucleus that you learn about. So you need to describe transcription, and it's worth 3 marks. So you need 3 key points. Okay, so here's the first 2. You need to say that the DNA unzips and unwinds, and the DNA bases are exposed. You might even say that the template strand is exposed, and it contains the specific sequence, or it contains the gene for telomerase. So this is the key here. We're actually mentioning the context that's happening. So always try and relate your answer back to the specific parts in the question. So there's your 2 marks there. And then for the third, RNA polymerase pairs RNA nucleotides with exposed DNA bases in a complementary manner. And we know the base pairing rules, but there they are for you anyway. The key there is that U pairs with A instead of T for mRNA, and it forms a single strand of mRNA. So the 2 key bits for this final mark are the complementary manner to form a single strand of mRNA.